Professor Rafi for the invitation to give a public lecture here. I'm trying to, hang on, I'm trying to share the screen. Can you see my screen?
Yeah, you can see, yes.
So is it in the presentation mode?
Yes,
Okay, that's great.
correct.
Okay. So very good morning, selamat pagi, salam sejahtera, everyone. I guess the time is 9 o'clock there and 10 o'clock in Malaysia. So as introduced by Professor Rafi, actually Lim. So surname is Lim, name is Vuanghao. So I'm actually the lecturer or the faculty of Pusat Kanser Tun Abdullah Badawi. So we have recently changed our name. Previously was known as Advanced Medical and Dental Institute. So we are actually based in Universiti Sains Malaysia, Penang, Malaysia. Okay, so this is my email contact, and I'll introduce a little bit of my research later on towards the end of the slide. Okay, next, the title of my presentation today will be From Plants to Health. So Metabolomics and Nutraceutical Discovery. So I believe that a lot of the members here are doing metabolomics. So today I'll be discussing about how untargeted metabolomics, some of the molecular networking, chemometric analysis, and also network pharmacology that can be integrated into a comprehensive platform for nutraceutical and also the drug delivery. So rather than studying one compound at one time, so these approaches could let us understand how the plant as a complex chemical and also identify compounds with a potential therapeutic value. Okay, let's begin with metabolite profiling. So we know that plants produce thousands of metabolites, and we know that many of them contribute to their medicinal properties. So we do have like traditional phytochemical studies, which mostly focus on the well-known compounds or the few known compounds. But the metabolite profiling, it actually allows us to investigate the entire metabolome simultaneously. So we know that this holistic approach basically enables us to compare. Compare what? Compare about different species, different plant parts, and also different environmental conditions. However, most importantly, it basically helps us to identify biomarkers and also some of the unknown compounds that may contribute to the activity. So before we discuss about the metabolomics, I think it is quite useful to understand how natural product research has traditionally been conducted. Now, for decades, we know that these are the standard flow that we can see involved in isolating compounds one by one before evaluating their biological activities. So we have the bioassay guided isolation, and next we do have the bioassay guided fractionation. Okay, so this figure we can see that it illustrates the conventional of what a bioassay guided fractionation approach, where some of the researchers, they will begin with crude extracts, then fractionate them repeatedly, then a refraction for biological activity. Then after this, they continue purification until a single compound is isolated. Although we know that this strategy has been extremely successful, but it is also very time consuming. It is very labor intensive. So some actually we look at as synergistic interactions among multiple compounds that present in the extract. So this limitation has encouraged researchers to adopt metabolomics-based approaches. So in terms of current status of plant metabolomics, as we can see that it has evolved rapidly over the past decade, and we could see that the advances in high-resolution mass spectrometry, computational analysis, and also some of the bioinformatics, basically they allow us to detect thousands of metabolites in a single experiment. Now, instead of asking what compound is present, we can now ask much more informative questions such as which metabolites differentiate one species from another, which metabolites actually correlate with biological activity, and what are the metabolite pathways are involved. So these developments have significantly accelerated natural product discovery. So when we come to the metabolomics, what is exactly metabolomics in plants? So it is a comprehensive of analysis of all the small molecules present in biological system. So these small molecules are considered the final product of the cellular processes, meaning they provide the closest representation of actual physiological state of an organism. And what more, it is to bridge the gap between the genotype and also the phenotype. So I believe that all of us know about history. So unlike the genomics and transcriptomics, so metabolomics directly reflects what is happening inside the cell at a given moment. And because they are molecules that are responsible for biological functions, analyzing provides them into nutritional value, medicinal value, stress responses, and also some of the disease mechanism. So of course, this is particularly very useful in the medicinal plant research. So in terms of combination of metabolomic in the system biology, what we can see here is the research does not stop at identifying metabolites, but we integrate metabolomics with pharmacology. So we do have the metabolomics tell us what are the compounds that are present. Their network pharmacology predicts how these compounds interact with multiple molecular targets and also the signaling pathways. So we know that this system level approach is especially suitable for herbal medicines because plant extracts usually contain hundreds of compounds that act simultaneously. So you could see that the system biology here, it shows that it flows from the RNA, proteins, and finally the metabolites. And among all the layers, the metabolites are the closest to the phenotype. So by combining the metabolomics and with the computational biology, we can connect chemical composition with biological function and better understand the mechanism underlying the therapeutic effects. So this is the so-called the combination of metabolomics and also the bioinformatics. So I would like to share some of my case study that I've done. So the number one will be Kanser species, which is a source of therapeutic compounds for breast cancer. Now, breast cancer remains as one of the leading causes of cancer-related deaths among women worldwide. Now, we know that although there are a lot of many treatment options are available today, several challenges remain, including the drug resistance, toxicity, and also disease recurrence. Then, of course, the natural products continue to be an important source for new drug candidates because they provide diverse chemical scaffold that are difficult to synthesize chemically. So, of course, this motivated us to investigate medicinal plants as a potential source as anti-breast cancer compounds. So, if you could see that some of the Kanser species and also the Rotunda species. For our first case study, we focus on these three plants, which include the Kanser galangal, Kanser parviflora, and also the Boinsaga rotunda. So what we can see here is all these three species are widely used throughout the Asia as the foods, spices, and traditional medicines. However, due to their popularity, comprehensive comparison, their metabolic composition and also therapeutic potential remains quite limited. So as you can see here, galanga is traditionally used as a digestive disorders and respiratory conditions. The parviflora, which is the black ginger, has attracted attention because of its reported effect of physical performance, metabolism, and also antioxidant activity. And the third one would be the finger root, which is very famous in Indonesia as well, especially in the cooking. So these traditional uses suggest the presence of diverse bioactive metabolites, which is worthy of further investigation. And of course, we could see that some of the flavonoids component that has been discovered. But if you can see that what are the differences between three of them and what are the similarities in terms of synergistic and also antagonism. So we did a very simple test. So I will start with the phytochemical content and also the antioxidant activities. So basically, we evaluated the total phenolic content, total flavonoid content, and also antioxidant capacity using the assays. The leaf generally extracts exhibited a stronger antioxidant activities compared to rhizome. So this is something that a bit different from what we thought. So suggesting that probably the leaves may contain higher concentration of antioxidant metabolites. So later on, we conclude that the initial indications where this sample should undergo a more detailed metabolomic analysis. So later on, we try with the anti-cancer activity, which is MCF breast cancer cells. So interestingly, we observe that several left leaf extracts demonstrated a stronger cytotoxic activity compared to their corresponding rhizomes. So among them, we can see that the Camphoria galangal leaf shows a particularly promising activity with lower IC50 values over the time. So these biological activities or biological results were later integrated with the metabolomics data to identify the metabolites that are responsible for the observed anti-cancer effects. So we did a simple PCA. So we can see that PCA, it shows the untargeted metabolomics is a unsupervised method where the metabolite variables, we can see that there's a distinct clustering observed among different species, which we could indicate that it's actually possess a unique metabolic profile. Then with the tight clustering of quality control, we see that demonstrate a good analytical reproducibility. So next, we proceed with the supervised learning, which is we use the PLSDA and also PLSDA models. And we put the breast cancer, which is an MCF7 activity as part of the Y variable. And observing the natural clustering from the PCA, we move to OPLSDA, where you can see that unlike the PCA, the OPLSDA maximizes the separation of Y separation between the predefined groups and help to identify the metabolites responsible for those differences. Then we can see in terms of biplot, the samples and also the metabolite features, they are displayed together, which we could see that metabolites are located closely to a particular sample group, which is highly associated with the species of the plant part. And also, for example, we could see that here, several methylated flavonoids, basically, they are quite strongly associated with the Camphoria perfusora rhizome. So we can see that these metabolites are known to possess antioxidant and also the anti-cancer properties, so making them interesting candidates for further investigation. So we did a S-plot and also the pathway analysis. So we could see that once the significant metabolites were identified, so the S-plot to prioritize the potential biomarkers, we could see that some of the metabolites that are located at the end of S-plot features that have both reliability and also high concentration or contribution to the sample discrimination. So there's a differences between the leaf and also the rhizome. And of course, the chimeferol emerged as one of the important metabolites. Then we then performed the pathway enrichment analysis, which highlighted the flavon and also the flavonoid biosynthesis as one of the major metabolic pathways. And of course, this finding is biologically meaningful because flavonoids are well known for their antioxidant and anti-cancer activities. Yeah, these two of them. Then we could see the volcano plot, which combines the statistical significance with the fold change. Appearing upper left and upper right corners, both basically they are statistically significant and substantially altered between leaves and also the rhizomes. So this will allow us to rapidly screen thousands of detected metabolites and also to identify only most of the biological relevant candidates for subsequent annotation. So of course, this is one of the essential filtering steps before go for compound identification. So from here, from all the values that we have obtained together with the VIP, we basically shortlisted several metabolites with favorable drug likeness and oral bioavailability. So we can see that the most interesting compounds were phenetidine, quercetin, isotretinoin, mosoflavone, ginidinine, and also epiloglycine gallate. So we can see that these compounds have previously associated with antioxidant, anti-inflammatory, and also some of the anti-cancer activities. So here, instead of relying on only one single compound, our integrated workflow basically enables us to identify a panel of promising metabolites that may work synergistically. So you can see that after the analysis of the whole thing, then these are the compounds that we have obtained. So what we're going to do with all these compounds, we're going to move to molecular networking. So molecular networking is a computational strategy that visualizes the MS-MS spectral similarity group structurally related metabolites. So at this stage, the identifying individuals' metabolites alone is no longer sufficient. So we therefore using the GNPS platform. So as you can see that the molecular networking group's metabolites, according to similarities in their MS-MS fragmentation patterns, in terms of rather than analyzing the compounds individually, we can also visualize entire families of structural related metabolites. So of course, this greatly improves the annotation efficiency and also provide insight into chemical diversity and also medicinal plants. Okay, so we have the integration with the activity as well. So in terms of the overview, as we can see here, if we did it in the positive mode, so we can see this slide basically shows a feature-based molecular network generating from the LC-MS data. And you can see that each node here, okay, each node represents one detected metabolite, while the connecting edges indicate structural similarity. So we can see that some of the clusters that have been formed based on the colors represent the metabolite families sharing their related chemical structures, and different colors correspond to different plant species and plant parts, so allowing us to observe which metabolite families are enriched in particular samples. So of course, this visualization provides much more information than a simple metabolite list. So we proceed with the positive mode on the flavonoid clusters. We're going to zoom in. So we focus especially on flavonoid-related clusters where we could see that flavonols, flavones, and also some of the flavonones, they are form distinct molecular families within the network. So you could see many of the compounds are recognized for their antioxidant and also their anti-cancer activity, for their ability to regulate the inflammatory and also cancer-related signaling pathways. So of course, this demonstrates that molecular networking can rapidly organize the complex metabolomic data sets into biologically meaningful groups. So we move on to the next one, which is some of the other cluster groups. So besides flavonoids, we also identify some of the numerous of other metabolite classes. So this includes the nucleosides, phenylpropanoids, and also some of the fructans. So although they receive less attention than flavonoids, however, many of these metabolites contribute to plant defense mechanism, antioxidant capacity, and also the immune modulation. Therefore, they should be not overlooked when evaluating the overall potential benefits of the medicinal plants. We also try to check other classes, for example, like these peptide-related compounds, and also some of the carbohydrate conjugates. So you could see that each metabolite class may contribute differently to the biological activity. So of course, this will highlight one important advantage of untargeted metabolomics, as it captures the complete chemical landscape rather than focusing only one group of compounds. So we can find that this is the amino acids, some of the carbohydrates, as well as some of the peptides together with their analogs. And not only that, we try to do with the lipidomics, which we try with the some of the maybe lipids, which are biological important molecules to the plant as well, and also to the biological activity. As you can see that the many lipids regulate some of the membrane integrity, cell signaling, and also inflammation. And also it provides some of the abnormal lipid metabolism that is frequently associated with cancer. So we can see that the same approach has been used. Then we can see in the positive mode, we see that there's some, this is showing the lipid molecular network, which is again, each node represent one lipid species, which connected nodes indicate structurally related molecules. So we are using the FBMNs using the same workflow. We were able to classify some of the numerous lipid subclasses and also to compare their abundance among different species and plant organs. And of course, this provides an additional layer of biochemical information beyond conventional metabolomics based on the different species here. So we could see that the molecular network has been colored according to the 6 different sample groups, as you can see here, and allow us to identify which lipid classes are enriched in specific species or certain tissues. So of course, such visualization basically helps us to understand some of the tissue specific metabolism and provides clues regarding biological functions associated with different lipid classes. We proceed with glycerolipids, which we have the glycerolipids classes. So among the lipid subclasses identified, we also have these glycerolipids represented by one of the largest groups, as you can see here. So this includes triacylglycerols, as you can see, as one of the subclasses, and also diacylglycerols, then monoacylglycerols, and also sulfur-quinol-diacylglycerols, okay, SQDS. So besides serving as the energy storage molecules, glycerolipids have been implemented in inflammation and also tumor biology. So you can see that their differential abundance may therefore contribute to the observed biological activities that I mentioned above. So moving on to other lipids. So we do have in addition to glycerolipids, we identify some of the fatty acids and several lipid metabolites. Some appeared as isolated single nodes, okay, suggesting probably unique chemical structures and did not cluster with other lipid families. And these unique metabolites probably may represent novel compounds, probably, I'm not sure. It could be that worthy of future structural elucidation. So this is the one that they have a single node. So after having this identified major metabolites, so the next question becomes, how do these compounds exert or show their biological effects? So this is where the network pharmacology comes in. Okay, so instead of for us to assume which one compound that acts as a one protein or as a one compound that is related to any biological activity. So network pharmacology here actually predicts interaction between multiple compounds and also multiple molecular targets simultaneously. So we integrate the metabolomics with the computational biology. We can basically construct a compound target pathway networks that will explain the mechanisms underlying antioxidant and anti-cancer activity. So this level of perspective basically is particularly suitable for studying complex herbal medicines and also therapeutic effects that often rise from the combination of multiple bioactive compounds rather than single active ingredients. Now, we do know that there are single active ingredients that what we can see in the pharmacy today, but we also do have some of the extracts that few extracts or few plant extracts that combine together as a indication for a certain therapeutic value. So nowadays we are moving into multiple bioactive compounds rather than single active compounds because we know that there are some other compounds that can interact synergistically to treat some of the diseases. So here we do have in terms of neuropharmacology, there's identification of potential targets, which will associated with the bioactive compounds from the list that we actually obtained just now. So also we do the construction of compound target pathway, and also we integrate between the metabolomics data with the biological pathway to elucidate the mechanisms of actions. So from here, we could see that based on the metabolites that are identified from our metabolomic study, we we construct the compound target disease network. Okay, so what we can see here, this network illustrates how multiple phytochemicals can simultaneously interact with multiple disease-related proteins. Now, unlike conventional drug delivery, where one drug will target one protein, natural products, normally we will try to show that their therapeutic effects through multi-target regulation. So we do see some of the several hub proteins here, for example, like AKT1, TP53, MAPK1, they are appeared to be repeatedly in the network. So we can see here, these proteins are well established in cancer biology. So suggesting that our identified metabolites may influence the key signaling pathways that involve in tumor progression. So from the sub, we try to do a protein-protein interaction network. So to better understand the relationship among these target proteins, we basically constructed a protein-protein interaction, which is the PPI network. We could see that from the proteins that we have obtained here, the highly connected proteins, basically we refer this as a hub proteins. So it's play a central roles in biological processes. Therefore, targeting these proteins may produce a greater therapeutic effects than targeting isolated proteins. So PPI here, analysis also reveals some of the connected signaling pathways associated with some of the cell proliferation. So in this case, it could be cell proliferations of the anti-breast cancer of MCF7, either be it in the apoptosis or maybe some of the metastasis. So providing a further evidence support the anti-cancer potential of the plants that I have mentioned just now. So from here, we have obtained that 6 compounds that could be as a bioactive compounds, as a multi-target bioactive compounds that could be responsible for the breast cancer. So next, we move on to the second plant, which is another second case study, where we focus on Clinacanthes nutans, which we call it as a Sabah snake grass. And this medicinal plant is widely distributed across the Southeast Asia and has been extensively used in traditional medicines. It used to be very popular in Malaysia and Thailand those days. Could be in Indonesia as well. Traditionally, it is used to treat skin infections, some of the insect bites, and also herpes infection has been reported in one of Thai journals, and also inflammations. And of course, more recently, there are researchers become more interested in potential anti-cancer properties. As you can see here, anti-cancer properties has been used a lot for this plant. So you can see there are a lot of reported papers on this plant as well for anti-cancer activities. However, most of them are using extracts. So active compound responsible for these biological effects remain incompletely understood. So we would like to target the NPC, nasopharyngeal carcinoma, since it is very famous in Malaysia, Thailand, Indonesia, and also China. So the MPC has a very high prevalence in China and also Hong Kong areas, which their percentage is actually quite high. So this is in collaboration with the Chinese university, which is Henan Medical University. So we try to find what are the compounds or bioactive compounds that are responsible for this MPC. So the characteristic of MPC is a very highly metastatic potential, even though that it is diagnosed at the very late stages, because it's like, the nose cancer or throat cancer. So the clinical challenge would be urgent need for a novel chemotherapy agent for a targeted molecular mechanism. So as I mentioned earlier, if you see the geographic prevalence, it is very highly endemic to South China and Southeast Asia. I'm not sure, probably because of the food or probably because of the way of eating. So in Malaysia, we have very prevalent in the Sarawak and also Sabah area. And of course, it is highly invasive. And now we use the cytotoxic drug with the cisplatin. Cisplatin has faced some of the tumor resistance, and also, of course, this will require a more novel therapeutic mechanism to overcome the MPC. So as I mentioned earlier, we do have the Suniten as a plant, and it has been used a lot in Southeast Asia. Some of the phytochemical compounds have been identified, for example, like those flavonoids, glycosides, glycerolipids, and also some of the cerebrosides. As I mentioned earlier, the pharmacological activity mostly on antiviral, anti-inflammation, anti-diabetic, and also some have been reported on anti-cancer. And in Thailand, this plant has been, produced in a dosage form, where they have the ointment, they have massage, they have balm, they have cream, they have lotion, and also they also come up with supplements in the capsules, and also some of them are packaged in the tea. So before beginning of any biological investigation, we basically ensure that the quality is actually essential. That's why the medicinal plant often exhibit a considerable variation due to geographical location. So we did a quality control in terms of the cultivation condition and harvesting time. So of course, the Metabolomics provides a powerful quality control tool because it evaluates the entire chemical fingerprint rather than relying only one or two marker compounds. So this results in much more reliable standardization of herbal materials, as you can see here. And different extracts were prepared, so evaluated for their biological activities. So you can see that the most active fractions frequently on these, some of the different places will show how biological experiments and computational analysis will complement each other throughout the discovery process. So quality control will see that different places will have a different variations. So this is how we standardize the quality control using the Metabolomics method. Then next, we engaged with the evaluation. Here we are using the CCK8 cells on the anti-MPC. Basically, we extract it into crude extract, which is the ethanol. This is ethanol water and also water. So we have the three crude extracts. What we can see here is ethanol extract consistently demonstrated a stronger growth inhibition compared to the water extract. So you can see that inhibitory effects also increase with both concentration and treatment duration, indicating dose dependent and time dependent activity. And of course, these results allowed us to prioritize the most promising extract for further metabolomic investigation. So from the three cell lines here, which we can see that ethanol water extract shows basically a good result. Then we proceed with the fractionation. We basically fraction it into a few fractions to further concentrate the bioactive constituents. So rather than analyzing the crude extract alone, we do have the fractionation simplify the metabolite composition, and which will improve the ability to identify compounds. So we did a heat map from the PCA score and also PLS by pot together with the permutation test. So basically it shows that some of the compounds are upregulated and some actually are downregulated. So depending on the plant extracts and also the compounds that are responsible for the particular activity of the MPC. And we could see here at this stage, the untargeted metabolomics perform on the active fractions, where we have some of the active fractions here, which we divide into polar and also non-polar metabolites, which is thousands of metabolite features were basically detected and processed using the MBDA. And this goal is to determine which metabolites correlated more strongly with the anti-cancer activity. So as you can see here from the fractions, we have the 1, 2, 3, 4, 5 fractions. We compared the metabolite profiles of fractions with different biological activities based on different timeline. So the fractions exhibited stronger anti-cancer effects clustered separately from the less active fractions. And you can see that DCM, which is dichloromethane. So dichloromethane of the kilonewtons, which is subarachnoid with the best anti-MPC activity. So of course, this shows very strong evidence that chemical composition is directly associated with some of the biological functions. Yeah, so these are we can see here based on the 3 cell lines. Basically, it shows that one of the most significant results. We did the metabolomics, which is the lower phase on that the 5 fractions, and you could see that from this core, quality control quite clustered tightly. PLS further demonstrates of clear separation among the fractions with different levels of biological activities. And also importantly, the permutation tests confirm that our statistical models are not overfitted, increasing confidence in the identified biomarker. So we see that separation of the fractions basically distinct the bioactive compounds or the markers or the metabolites from different fractions. So from the whole list, we found the candidate of bioactive compounds. Of course, using the annotation, we identify several metabolites that are highly associated with anti-cancer activity. In this case, is MPC. So these compounds became our candidate of bioactive metabolites, is many of them, but we are trying to scope it down. So rather than claiming that one compound is solely responsible for the observed activity, so we can recognize that there are multiple compounds may contribute synergistically to the therapeutic effect. So we go for the target prediction. So once the candidate compounds were identified, so we predicted their molecular targets using this Swiss target prediction and also together with the TCMSP database. So this computational approach basically estimates that which human proteins that are likely interact with the identified phytochemicals, as I mentioned earlier just now. So you could see the resulting compound target network provides a valuable hypothesis that can later be validated experimentally. So you can see that there are a lot of proteins that have been targeted in terms of this case, and the network relationship of the screen compounds together with the targets. So we try to do a disease target prediction. So the next step is to identify which genes are basically associated with MPC, which is the nasopharyngeal carcinoma. Then we collected the disease-related genes from multiple geo database with the transcriptomic datasets together with the disease databases. So we combine some of the several independent datasets to improve reliability and reduces bias associated with any single study. So you can see that the overlapping. So some of the overlapping disease-associated genes and compound targets basically form the basis for identifying potential therapeutic mechanisms. So you can see that there are some of the volcano plots that are showing upregulated and some of that are not significant. We proceed with the PPI network. So after identifying the disease-associated genes and the predicted targets for our phytochemicals, we look overlap between them. So we see what are the genes that overlap that we can scope it down. And these overlapping genes represents the most likely therapeutic targets to clinical newtons. The plant itself may exert some of the anti-cancer activity. Then the protein-protein network interaction to determine which protein occupy the central position within the network. Okay, as you can see here, these are the top 10 hub genes that have been identified. So these hub genes that are identified are likely to play a critical role in nasopharyngeal carcinoma progression and therefore represent promising therapeutic for future validation. We then proceed once the core target has been identified. Then we did perform some of the functional enrichment analysis. As you can see that this analysis tells us about biological processes that happen and also some of the signaling pathways that are more significant associated with the predicted target. Rather than affecting one single pathway, these results may suggest that multiple cancer-related pathways are simultaneously regulated. So, for example, like this multi-target characteristics is one of the major advantages of natural products compared with conventional single-target drugs. And this KGG pathway provides another level of biological interpretation where several important pathways involved in cancer progression, apoptosis, inflammation, and cell survival were significantly enriched. So you can see that some of the pathway that can show cancer pathway, also cancer progression. So these findings suggest that the phytochemical identified through these metabolomics may influence the multiple signaling, which is some of the plausible explanation and the observed anti-cancer effects. So from here, we found that the whole figures are the information in the single network. So on the left here, you can see there are bioactive metabolites that are identified from the metabolomics. So we have the Canadin B, josecidin, epigenin, sinacetin, and some other compounds. So these are, in the middle, we have see that these are the predicted molecular target. These are the targets from these compounds that have been, bioactive compound that has been identified. So on this thing are the biological pathway associated with these target genes. Then we can see that this system level network demonstrates that multiple phytochemicals contribute to the multiple signaling pathways simultaneously, and this will explain why metabolomics and network pharmacology complement each other to effectively in natural product research. And of course, we try to use a computational predictions alone are basically not sufficient. So we need to perform a biological validation. Okay, biological validation. In this case, we just use in vitro assays, where we try these experiments to demonstrate whether our active fraction significantly inhibited cell proliferation or not. So, and also we did some of the migration study, while reducing some of the colony formation in the nasopharyngeal carcinoma cells. So we could see that this experimental result strongly supports a prediction that generated by the metabolomics and network pharmacology analysis. So it's already proven, validated. So from the metabolic study and also network pharmacology study, we validated with the cell study, and it says, oh, okay, yes, it is actually working. Then next, we did cellular metabolomics. So just now we did on the plant extracts. Now we did the cellular metabolomics, which is the metabolomic in the cell. So after confirming the biological effects, we also investigated what actually happens in the cancer cells after the treatment. Okay. So instead of analyzing the plant extracts, we analyzed the metabolic response of the cancer cells themselves. So we could see that there is a clear separation between the treated and also the untreated groups, which indicate that the treatment induced significant metabolic reprogramming within the tumor cells. Okay, so we actually tested with epigenetic because actually epigenetic shows a very high significance on the MPC. And this provides a direct evidence that biotic compounds in terms of the epigenetics are influencing the cellular metabolism. So you can see that the treatment group and also the control are well separated in terms of the compounds that has been expressed. So we checked the identified metabolites that changed significantly on following treatment. So across the three MPC cell lines, there are numerous metabolites show significant alteration. Okay, so some metabolite changes were common across all three lines, where others were cell line specific. So it depends on the cell line itself. And these altered metabolites provide a valuable insight into biochemical mechanisms through which the plant extract, in this case of Clenacanthus nutans, show its anti-cancer effects. So this may serve as a potential markers for a treatment response in the future. So to better understand that whether these metabolites this metabolic alteration, so we constructed the compound reaction enzyme gene network. And unlike the previous network pharmacology analysis, where we started with the plant compounds, so this network starts with the altered metabolites detected in the treated cancer cells. So by treating, integrating the metabolites, enzymes, reaction, and also some of the genes, we obtain a much more comprehensive picture of how treatment that affects the cellular metabolism. So this system biology approach basically allow us to identify key regulatory nodes that may be responsible for the observed therapeutic effects. So we also see some of the compound reaction gene as what we constructed here. So we could see some of the metabolism. So metabolism of what? Some of the metabolism of tyrosine, urea cycle, metabolism in the cell itself, and also methionine and cysteine metabolism. Okay, valine and leucine, and also pyrimidine metabolism from the whole picture of what I have mentioned just now. So for the key findings, to conclude today's sharing, I would like to highlight some of the several important to take home messages. So the first is untargeted metabolomics enables comprehensive chemical profiling of medicinal plants, which allow us to move beyond the traditional one compound at a time approach. Then secondly, to the molecular networking significantly improves the metabolite annotation by organizing structurally related compounds into meaningful molecular families. Then third, we do the integration of metabolomics with network pharmacology, allows us to bridge plant chemistry with disease biology by predicting molecular targets and also some of the signaling pathways. And finally, by combining the experimental validation with computational analysis, this will provide a powerful strategy for accelerating the discovery of phytochemicals for nutraceutical and also pharmaceutical development. So overall, it demonstrates that metabolomics is no longer just an analytical technique, but it also becoming a central platform for modern natural product research and also precision nutraceutical discovery. So yeah, and here is the my group, which we call it an drug and metabolomics research group. So these are all my students, PhD students. Some are abroad and some are here in Malaysia, and some are my postdocs. Would like to express my gratitude to them, because without them, I mean, this work couldn't be a successful one. And also special thanks to my collaborators. So we do have collaborators from different parts of the world. We do have collaborators from Indonesia as well, from the locally, from the US, from the Australia, Singapore, and the UK, so on and so forth, and so in China. Then a little bit introduction about my institute. So if you can see that this is the map of Malaysia. So we are actually based in Penang, which is and a very small island. So we have 3 campuses in Peninsular Malaysia, which is the Georgetown, which is the Penang. Then we have one engineering campus in the southern of Penang, and our institute is actually at the north of Penang mainland. So it is called the Advanced Medical Analysis those days. Now we have changed our name, Pusat Kanser Tun Abdullah Badawi. So it is located here. And yeah, so this is the pictures of our main buildings. And yep, thank you for your attention. So this is my profile. If you would like to collaborate anything, you can just scan the QR code, and I'm happy to collaborate in terms of research collaboration and also whatever collaborations. Thank you for your attention, and I'm happy to answer any of the questions if there is. Okay, let me see. All right. Thank you very much.
Yeah, okay. Thank you very much, Dr. Lim, for the nice presentations. Anyone for the questions from the audience? Just please raise your hands or just write the questions, yeah, in the chat room. Okay. Anybody? Yeah, okay, doctor. Maybe from me, yeah. Okay, there is someone. Please, Reti.
Thank you very much, Prof. Raffi. And thank you for the very insightful presentation, Dr. Lim. My name is Reti, and I have one question related to the network pharmacology. As you know, that since many medicinal plant has activities has reported to have an activities like anti-cancer, and my question is how did you determine what the specific disease in this case is breast cancer and NPC, right? From the broad pharmacology activities like anti-cancer. That's my question.
Okay, thank you for your question. I'll try to refine your question. Let me see if I understand your question correctly or not. So you're actually asking that how can one use neuropharmacology to determine the compounds for anti-cancer to determine the compounds for breast cancer and also NPC in this case. Is it correct?
How did you choose the disease, breast cancer and NPC?
Okay. Thank you for your question. Very good question. How do we choose the disease? So basically, of course, we have to do a little bit of literature background. So some of the ethnopharmacology of that particular plants that have been reported that this plant is actually very good for a certain disease. Of course, from the uh, previous studies and also from the literature search. So we found that, oh, this is actually quite potential maybe for breast cancer activity, because there will be some of the plants that they normally reported a very general, maybe for just cancer, but it is our duty, it's our responsibility to check what kind of cancer that these plants can be useful in, or can be useful in treating a certain part of cancer. So in this case, like the ginger, the camphorous species, of course, it has been used a lot in our food, in our medicinal properties in Southeast Asia. So some of the has been reported in literatures, and of course, we need to do the validation studies. Same goes to the clinic and the Newtons. Yeah.
It's okay.
Thank you.
Yeah.
Thank you, Dr. Lim.
Oh, Okay, doctor, there is one question also in the chat room. I will read for you. Since computational metabolomics and network pharmacology depend heavily on software algorithm and mathematical modeling, how reliable are their predictions? What inherent limitations lead to bias or discrepancies when comparing these in silico results to real physiological processes?
Okay, I'm trying to read the questions.
Yeah, in the chat room.
How reliable are their predictions? Yeah. Okay. So yes, correct. These is some of the discrepancies lah that we say that lead to bias and comparing in silico results. That's why when we do, that's why it's an interconnection of what? Interconnection of from the metabolomics into the network pharmacology or molecular networking, then to the network pharmacology. So when we try from the metabolomics, we are trying with some of the validation tests, which is a permutation test to see whether the model is correct or not. Then from the group of data or the metabolites that have been found, normally we will try to test it with the in-house library or with the reliable database. Then from this group set of data, we test the drug liners and also some of the potential target in the network pharmacology. Then we proceed into the activity correlation, which is the what we call it as targeting of proteins and also targeting of the pathway. Then of course we need to validate it back. Of course, when let's say we have the 6 compounds or 6 metabolites, we have to check back with the HPLC together with the main compounds to see whether they are actually present in your extract or not. Okay, if they don't present, that means there is something bias there or there's something wrong there. So we have to check back to the HPLC. If the HPLC shows that the existence or the presence of your bioactive compounds, then we will test that bioactive compounds into the activity again, which means that we revalidate it. As I mentioned in my slide just now, we actually revalidate it and to check whether that activity will be the same as what has been reported by the bioinformatics processes just now. So if it is validated, then of course we have to check what are the responses of the metabolite programming and also some of the processes and signaling pathway. Yeah. So validation is very important. Of course, I do agree that computational metabolomics and network pharmacology will depend on the software algorithm and it will give some of the discrepancies and also biases. Validation is very important. Need to revalidate it back. Yeah.
So you mean that some isolations of the predictions compounds is very important also as the validations, yeah?
Yeah.
Mm.
If let's say some of the compounds that have been identified, you actually can get it from the market, then you just get it from the market. Otherwise,
Yeah.
you can just focus on that particular compound, you just isolate it and just retest it. Yeah.
Yeah, especially if there is no reference standard, yeah, for
Yeah,
this
if there's no
compounds.
if there's no reference standard, then no choice, then you have to isolate it. But of course,
Yeah,
we
yeah.
are not targeting on a single drug. We are targeting on the multi-target drug. Yeah,
Multi components, yeah.
yeah, multi-components.
Okay, maybe one question for me, Doctor, regarding the
Yeah.
annotations of the metabolites. Sometimes because we use the databases, yeah, we lead to the misannotations. Yeah. So, what would you like to guess to have correct annotations of our metabolites from the chromatograms with the mass spectrums, so we can make sure that our annotations is will be similar with the chemical structure of the predicted compounds.
Okay. Yes, annotation is very important. If let's say we are doing annotation with the in-house, definitely we will get the very correct one. So in this case, we have the confidence level lah. So we have level 1, 2, 3, 4, as we all know. Identified compounds, we can test with the the standard compound. If we don't have, then probably we need to do a putative annotated compound. Then we need to test the MS. The MS we can actually basically check from the databases, and otherwise we have to elucidate in ourselves, which means that we have to check the fragments, we have to check the MS fragments. Then from there, we have to check with the established database. Otherwise, if we really couldn't find it, then we can get the cluster from the molecular networking. Then we will see what are the family cluster that is closely to the one identified. Then we need to check their MS of the cluster family. Then that MS, we need to do a further check. Further check as in like either you do it manually or you do it with the website. Yeah. With the database.
Yeah. You mean that we cannot merely 100% trusting from the machines coming out with the predictions of the structure. We have to confirm with like fragmentation or something in our databases.
Yeah.
Okay.
Unless you have an in-house library or unless you have the standard
Library,
compound,
okay.
yeah, that would be a different story.
Yeah, if we have reference standard, just running with the reference standard, right? Yeah, so we compare the fragmentations pattern. So I think this is the most also the important things, one of the biggest problems, in our metabolomic study for the annotations. Okay, there is any questions? Okay. Would you refer where I can read the whole workflow of the research in your presentations? It is really comprehensive. I couldn't catch up. Thank you, Prof. Lim. I think you can still hear in the YouTube from Forum Metabolomic Indonesia, Ibu Septiari, So, and I think in the Dr. Lim presentation, there is some citations, Doctor?
Yeah.
You can download the paper or something regarding the Dr. Lim works, I think. Or you can add more information, Dr. Lim.
Yep, can do. I think you can also check one of my papers. Some are unpublished papers, so probably some published papers, then you can just refer to the published papers. You can just go to my profile, Google Scholar, then you can get the information there, the publication. Yeah.
Okay. Any more questions? Pak Hong ada mau tanya? Yeah, this is one of my friends from Universitas
I'm so
Jenderal Ahmad.
sorry. Thank you,
Okay.
Prof. Raffi. Prof. Lim,
Yeah.
thank you very much for your insightful presentation. Actually, I'm not a fully metabolomics. Mostly, I work on the proteomics part and protein engineering mostly. I was wondering, actually, just assumption, if we have a set of compound in particular herbs, for example, or plants, and then can we use the set of the compound for the molecular docking in order to find the active compound which actually work on the particular, let's say, therapeutics activity, or we have to determine directly from the plant without any without any experimental experiment to use your workflow. Thank you, Prof. Lim.
Okay, thank you very much, Prof. Khomeini. It is a very interesting question. Yes, from the compounds or the metabolites that you have identified, bioactive, that means you have scoped down, you have these probably about 6 to 10 compounds that have behind that can be considered as a bioactive compound. You can actually use that compound to do molecular docking. Yes, I do have the molecular. We actually did the molecular docking, but I didn't present it today. Yeah, so yeah, you can actually do the molecular docking and check the prediction target and also check whether these compounds can be used or can be as a potential as a target for that particular bioactivity that you are trying to test it on. Yeah, can do molecular docking. We actually did that molecular docking and actually tally with the result from our validated data.
Okay,
Yeah,
clear. Thank you.
thank you.
Okay, thanks, Pak Khomeini. Yeah, thanks. I think, is there any more questions? If it is not, I think thank you very much, Dr. Lim, for your comprehensive talk this morning. Yeah, hopefully, yeah, you also had some collaborations with university, especially in East Java. Yeah, East Java, because I
Yeah,
just
sure.
know the logo from Brawijaya and the Technology 10 November and
Airlangga.
also Erlangga. Yeah.
Airlangga, yes, Airlangga.
Yeah. Now is my daughter pursue her bachelor degree in Brawijaya University.
Oh, okay, that's good. That's good.
Yeah. Okay. But in the Department of Chemistry, yeah, same with
Great,
me.
great. I just went
Uh,
there. I just went there about 2 months ago.
2 months ago.
Yeah,
Oh, which faculty you
back to
went?
dentistry.
Dentistry. Oh,
Yeah.
dentistry. Okay,
Yeah.
okay. Okay, dentistry. Okay, maybe later you can go to other places in Indonesia. Also, I would like to inform you that next year we will have Indonesian's Metabolomic Symposium, the third one. Maybe you can come. I will inform you about the symposium. Yeah.
Okay.
Also, now in Indonesia we have the associations for the Metabolomics Society now.
That'll be
Yeah.
very good.
Very much, Dr. Lin. See you offline if can be. Yeah.
Yeah.
Now also I'm in KL.
Oh, which part of KL?
I'm doing my sabbatical leave now in the University Malaya
Oh, you
until
will.
the next August. Yeah.
Is it on metabolomics as well?
No, in the analytical chemistry because my background is analytical chemistry and I develop methods for the metabolomics.
I see. Okay, good.
Yeah. Yeah, I meet also one person from the USM that you have also collaborate, Dr. Martiana
Oh, for the
in
one year.
Jakarta,
Okay.
yeah, last year, I think.
Okay.
Okay, so I know you from her, yeah. So I contact searching
Okay.
for you to give this nice lecture from you. Okay, thank you very much, Dr. Lin,
Thank you
for
very
your
much.
kind sharing. Yeah, see you again. Thank you very much for everyone. Ya, saya akhiri untuk kuliah umum metabolomik ke-63 ini pada pagi ini. Insya Allah kita bertemu bulan depan di kuliah umum metabolomik ke-64. Ya. Thank you very much.
Thank you
See
very
you.
much. Okay.
Thank you so much, Dr. Lim. Dr.
Terima
Lim.
kasih, Pak Raffi.
Thank you, Dr.
Sama
Lim.
Pak
Thank
Terima kasih
you.
juga sudah
Terima
hadir.
kasih, Pak
Terima kasih Bapak dan Ibu sudah hadir.